It's my great pleasure today to give this lecture to you about early innate immunity and lung development. Respiratory viral infections which result in wheeze, shortness of breath, respiratory distress are the most common cause of hospitalization in children below the age of 5. And the viruses that mainly cause wheezing in children are the RSV virus, the respiratory syncytial virus, as well as the human rhinovirus. Both of them infect the patient via the upper respiratory tract and infect not only the nasal mucosa, but also mainly the trachea and the epithelium of the bronchi, the bronchial epithelium that you can see up here. Mainly these epithelial cells are infected, which are our first line defense against viruses and can themselves, once infected already, act against them. However, of course, they also need the help of the immune system, which then infiltrates the epithelium. Here you can see the diversity of the immune cells that infiltrate from below, from the basal side, but there are also cells that actually transmigrate and sit on the apical side of the bronchial epithelium. Here you can see an electron microscopic picture from the thumb, where you can see an alveolar macrophage that scans the surface of a bronchial epithelium. Here you can see actually the cilia of the bronchial epithelium, and this is the macrophage. But not only macrophages, as part of the innate immunity, are residing within the lung, but also all the different other cell types of the innate immunity, comparison to the adaptive immunity we have here. Monocytes in general, phagocytes, dendritic cells, mast cells, NK cells, also novel innate lymphoid cells, ISCs. And the main feature of the innate immunity is that they react very fast. As you can see here, they migrate to the site of infection within 1 day or half a day, within the first hours, but they react unspecifically to the pathogen, which is in contrast to the adaptive immunity, which then comes in later and specifically attacks the certain pathogen. The wheezing children that I just talked about have a high risk to develop asthma. Here I show you a graph from the German Center of Lung Research. And we were particularly interested in general in the early age. What happens with these 30% of all children who develop wheeze within the first 3 to 6 years, so preschool age. It's a huge number of children. Most of them, or a big part, loses this wheezing, fortunately, and so they resolve. However, 15% of these children keep this phenotype, unfortunately, and develop persistent wheeze and atopic asthma, which culminates in approximately 200,000 children in Europe per year that in the end develop a bronchial airway epithelium or actually a airway wall, as you can see here in this microscopic picture, with a huge increase of smooth muscle cells and a constriction of the bronchi. A huge undertaking has taken place or is taking place each day worldwide to try to decipher molecular mechanisms, disease-driving mechanisms. These are only a few of the children cohorts worldwide. And I would like to point out one study around Dr. Dandl, who is also so kind to join us here today, that actually has a cumulative patient number that I have not seen before of 150,000 children. And in this cohort, they looked at lower respiratory tract infections and their association with reduced lung function. You can see here that these children that in early life developed lower respiratory tract infections actually have a decreased lung function, in particular FEV1, and here you can also see the Tifino index throughout the early years. And I can tell you that this lung function does usually not get better Yeah, so if you take the wrong path, this is what I will conclude with, then you might not get the perfect lung function you would like to have as an adult in the end. These children also are at a higher risk to develop asthma. As you can see here, this is again from the same meta-analysis of these cohorts, in particular, as I mentioned before, also in association to RSV infections. So the red ones had RSV infections in infancy. The percentage of current asthma at 5 years is much increased. Also, the percentage of developing wheezing episodes during the early ages. The first time when lung function was recorded, it looked like this. So you have this huge balloon in this little baby. 1952 in Stockholm, the first recording of the first breaths of a baby were performed by Professor Karlberg and nurse Kerstin Werner, as you see here, at a so-called mingograph, which spit out these kind of curves. Back in the day, the ultima ratio for children, in this case with poliomyelitis, who were not able to sustain breathing by themselves, was the iron lung. And the iron lung came back to our minds recently, within the last now 4 years of time, during the COVID-19 pandemic, in particular, when you look at one patient who has survived, I think, 74 years within this iron lung, but then unfortunately died of COVID-19. Our ultima ratio now for these patients was the ECMO. However, as you all know, due to the high number of patients, we also had huge problems with the amount of ventilators back then. But I would like to show you this to make everyone aware of how important it is to push this respiratory medicine field forward, that these innovations have brought us such a long way from the 1950s to today. COVID-19 taught us a lot of things. And in regard to the RSV infections that I've shown you just now, or that I've talked about, I would like to mention a study from Dr. Maison, who was also so nice to join us here today, and which showed that children in 2021 have a steep increase in RSV infections. This is now the data from RSV. It's also the same for the rhinovirus. But if you compare that to children from 2020, 2019, 18, 17, they didn't show this increase. Why are these children in 2021 all of a sudden unable to defend themselves well against respiratory viruses, in particular following the lockdown due to COVID-19? The current hypothesis is that they lack training of the innate immune system due to social distancing. If you think about the lung development, you mainly think about this part. This part happens in utero, starts with the embryonic phase, then after week 7 of gestation, you start to see the development of conducting airways during the pseudoclavicular phase. During the canalicular phase, then get the first primitive alveoli, then you go into the saccular phase, and if you're lucky and the child is born at 30 weeks, then you are already in the alveolar stage. But if you're not, if you're before week of 36, they're still in the saccular stage, meaning that preterm children actually don't even have the full alveolar system. But on top of that, today I would rather like to focus a little bit on the postnatal part of the lung development. You can see here that also if the, children are born at week 40, that there's still an increase of alveoli over age. So the lung development, in particular the continued cellular proliferation and lung growth, still continues after birth. Let's have a short view back in history. Which animal or plant, or probably animal, had the first type of kind of like a lung? I think most of you now think of amphibians, and you're right. So the first lung-like organ was actually, or was developed pretty much, or had this little guy. This African horned frog was one of the preterm frogs that had a lung-like organ that was constituted of one air sac. In general, amphibians have these air sacs. The salamander you see has already two. The lizard actually started now to have multiple chambers. The bird had multiple air sacs, but only the mammal in the end has a lung-like organ like we know it, with the bronchial diversification and the alveoli endings. When you now think about what I would like to talk to you today to bring together the innate immune system that sits within the lung and the lung, do you think they're older or younger? So the adaptive immune system is older. The first RAG-like, so recombinase activating gene-like genes were expressed in the sea urchin. But the innate immunity actually is much, much older than all the rest of them. The first toll-like receptor, toll actually coming from the German word toll. So these awesome receptors were first expressed in the sponge. In the prehistoric sponge, we have the first TLR, which had a homology with human TLR6 and TLR1. Why am I mentioning this? Because these awesome receptors are a critical part of the innate immune response and also of the trained immune response I would like to touch upon quickly. They are so-called pathogen-associated molecular patterns recognizing receptors, so PRRs that recognize PAMs. That means that they recognize particles of bacteria and viruses. Bacteria mainly recognized by TLR 1, 2, 4, 5, 6, and 9, while the viral particles are rather detected by TLR 3, 7, and 8. And what you see here is that these three receptors that I would like to focus on now, that these induce interferon regulatory factors and thereby the expression of interferons. Interferons are crucial molecules that are expressed following a virus infection, depending on the type of interferon, either in the infected cell, if you're looking at type 1 or type 3 interferon, so interferon alpha, beta, or lambda, are expressed right within the infected cell and are able to start or to trigger the expression of antiviral factors within the cell that, for example, might dissolve the viral genome, for example, RNases. They are not only acting on the own cell autocrine, but also warn the adjacent cell in a paracrine mode. On the other hand, we have type 2 interferon, which is also an interferon, but it's mainly expressed by infiltrating immune cells by the IC1s, TH1s, and CTLs. These interferons are, however, not only important for the spontaneous immune response, but they can influence also epigenetic changes and metabolic reprogramming of M1 macrophages towards glycolysis and ROS. Why am I, or reactive oxygen species? Why am I mentioning this? Because the trained immunity concept says that if you have an initial response due to a primary challenge with a pathogen or even a vaccine, you have this kind of small or short strength of an innate immune response. Then you have a training window, and during this training window, you need exposure to other pathogens in general. Let's put this in general. If this training happens, there's a metabolic rewiring and an epigenetic rewiring of the innate immune cells happening, which then lead to a much higher strength of the trained innate immune response after a secondary infection. If you don't have that, then you don't only lose the strength here initially, but you don't come back to baseline even. And the peak that you would like to see, for example, in these children that get RSV, you don't have. This is at least the concept We are in the very early stages, but this is the concept how we explain these tremendous increase of infections following lockdown. During the immune cell development in the lung, there are a few cells I would like to mention. The embryonic macrophage is crucial for the fetal lung morphogenesis. In particular, it secretes cytokines that leads to angiogenesis and to the building of the alveoli, of the branchings and so on. But you do not would like to excite these macrophages. An activation is unfavorable and is associated with preterm birth and perinatal death. The fetal NK cells, however, at least what the evidence says so far, can respond to infections in utero and are not associated with a bad outcome. In the canalicular stage, you have the surfactants proteins are being started to be expressed, and particularly surfactant protein A prevents the preterm birth by inhibiting these pro-inflammatory TLR signalings. In the canalicular stage, you also get the infiltration of fetal liver-derived monocytes, which then, during birth or following birth and the exposure to air, migrate into the lumen here and develop into alveolar macrophages, our really first-line defense, because as I showed you, they sit on top of the epithelium. The antigen-presenting capacity of dendritic cells is most probably reduced, in particular because we have a regulatory environment. PD-L1 is expressed on dendritic cells, and we have an influx of natural Tregs. The naive T cells that appear in newborns have a Th2 bias, meaning if they are activated, they rather tend to differentiate towards Th2 and tend to express Th2 cytokines. IgA, on the other hand, an antibody that we would need that can cross the epithelial barrier and can defend already initially, is only there after the first year of life. Important here is that the exposure to airway microbiota that starts to popularize the airways here following birth, during the normal usual birth process, or also due to breastfeeding, they start to populate here the airways, and this exposure leads actually to the training of these monocytes. This exposure, due to the hygiene hypothesis that has initially been brought up by Frau Professor von Mutius, is necessary for healthy development of the maturing immune system. In particular, we have association with the mode of delivery, with family size, SIP-SHIP, daycare attendance, and also with close contacts with pets or farm animals, in particular, classical farming. If the mother already is performing classical farming, this actually prevents the development of allergies and asthma. A lack of this exposure, however, increases the susceptibility to viral infections and allergies. And this microbiome can be composed of different bacteria mainly. The key driver of this microbial maturation is the gestational age at birth, the mode of birth, type of feeding, and the traditional farming that I mentioned. You see here approximately an overview of the different types of bacteria and microbes that you would like to have in a healthy microbiome that lines the airways. However, in a diseased one, we have a shift towards sometimes one or the other species. Inhibitors of this microbial maturation, therefore, are infections, but also the courses of antibiotics in very small children can lead to a dominance of some of these bacteria. Two of them are associated with wheeze and asthma. Staphylococcus aureus dominance is associated with wheeze, and the appearance of Moraxella in any subtype, but mainly Moraxella cataralis, is associated with asthma, in particular in association or in the combined with RSV infections. To sum up, we need training of the innate immune system by exposure to microbes postnatally in early life. Factors are, as I mentioned, natural birth, breastfeeding, siblings, daycare, farming, and no social distancing. These are positive factors. This training is necessary for healthy development of the maturing immune system, for the prevention of allergies and asthma, and therefore, simply said, I have to say, for a healthy lung development. This is what I mentioned in the beginning. And lung, either you go this lung trajectory or this over the age, you see this here, you don't catch up. They don't really catch up. So last but not least, if we dream or if we look into the future, what's our vision? Our vision is to identify children to treat in the window of opportunity to prevent the development of asthma. Our obstacles are scarce pediatric evidence of age-related immunity. The accessibility, of course, in small children of biomaterial is difficult and ethically challenging. The continuous longitudinal sampling is difficult. Some biologics we would think might be helpful in the early ages are not yet approved for small children. Our needs to overcome these obstacles are biomarker matrices to identify children at risk. Also, then we need to identify the age window of opportunity. So when should we treat? And then most importantly, we need early medical information by pediatricians, in particular of small children and their parents. We need home care, including digital tools like apps. We need easily accessible biomarkers, for example, nasal secretions, and we dream of a new drug for the early prevention of asthma. And I think this is where I would like to close. As you all know, I've spent 1.5 years in the United States. And what do the Americans say about the upbringing of a child? They say it takes a village. It also took a village to finish this habilitation. And therefore, I would like to thank each and every one of you, in particular, Carsten. Carsten, I would like to thank you very much for your support, your mentorship. Also, Adam, it's just very important. The whole group, Madeleine, Ferdinand, Helen, Shohung, Anna, Lynn, Lisa, Jana, Stephanie, Steffi, Marie, Amre, Ulrich, Emily, and Lucy, but also Simon, Francesca Alessandrini, Jeroen, Cordula, Miriam, and everyone else, of course. Yeah, so the list goes on. All colleagues from Zaum and HNO. Of course, the Fachmentorat, Carsten, Professor Biedermann, Clarissa, thank you very much for accompanying me on this way. The Haunersche Kinderklinik and the Helmholtz Center Munich with Professor Erika von Mutius, Professor Bianca Schaub. As I mentioned already, Nicole, Sabina, and Jimmy, Marie, of course, then Benjamin Schubert, Maren Büttner, thank you for being online. This is very nice of you. And Felix Trost, but also, of course, we had also a huge consortium together with Professor Protzer, with Martin Feuerherd, also with Professor Pichelmeier and with Dr. Vincent Grass. I would like to thank the whole DZL. This doesn't fit on the page. All patients and parents, all colleagues, all of our consortia, and all participants. In particular, I would like to point out the Advocate Consortium, Professor Procco, who is also here, and Valentina Feis, and also Dr. Marie Standl, of course, and the Graduiertenkolleg under the leadership